Ding dong. Hello, people. How many people we got today? 20. Let's wait a couple minutes because look at how I keep fading out. Whoa. Turn the light on. I am a ghost. Oh, well. I'll just have to be ghosty today. Where are we? We're up to 23. Got your dog. Can we put our faces up so we feel like a class briefly? Stefan. Nice. Antonio Estevez. So, Antonio, are you related to, oh God. No, I know you're not. I'm trying to make a joke. Emilio Estevez and his brother is the crazy guy from Three Men and a Baby, Two Men and a Baby. I forget. Oh, what's his name? I don't know. Never mind. Never mind. I can't remember his name. But there were three famous actors from my era named Estevez, although they changed their last names. Two of them did, one of them didn't. They were the guy from West Wing. What's his name? The guy who was president in West Wing. My brain's not working today. But anyway, his two sons, one was Emilio Estevez, and the other one was, oh God, the guy from Platoon. And he was in Two and a Half Men. I don't know. He was in Two and a Half Men until he decided he liked cocaine more than acting, and that was the end of that guy. Anyway, where are we at this point? 26. 26. 3 minutes after. All right, let's just go ahead. We are at 27 out of 34. That's not so bad. We're only missing 7. The cool kids showed up, right? All right, so first thing I want to do today is back up a little bit. So I've been talking about differentiation and cell fate changes and self fate mapping and all this stuff, but I don't think I really explained what that means and what we're talking about here. And it struck me that when I was reading the textbook today, and I saw this statement here, and I'm like, yeah, of course. And I thought, well, did you ever tell them that? Probably not. So I thought I'd just back up and talk about this today. So from the book I read, the state of determination of individual cells or of small regions of an embryo is studied by transplanting them to a different region of the host embryo and seeing how they develop. If they are already determined, they will develop according to their original position. If they're not yet determined, they will develop according to their original position. So determined, what does determined mean? We haven't really discussed that. It has to do with cell lineage stuff. So this is from a paper, which you can see the link there. It's about how certain ectodermal cells will go on to form the skin of your face and your teeth and other parts of the face. So hang on one second. I got to close the door. My wife's vacuuming. So the idea is somewhere during development, these cells form from ectoderm. Now these cells will then be stimulated by some hormones and then become either these cells or these cells. These cells will then can then become either these or these. And then you've got kind of stem cells here that will go on to become these 4 different types, or these 6 different types if they came from here, these 3 from here, and so on. So the basic idea is when these cells go down this pathway, they go from being up here, pluripotent, this cell here can become any one of these, to becoming more and more determined. And by determined, it means it's losing possibilities and becoming more and more specific. So if we follow this pathway, this guy here, once you get to this point, these cells are no longer able to make any of these cells or these cells, is determined to become one of these cells. And this is really what the process of development is all about, turning different genes on to make different proteins and going down a path towards some eventual adult cell. None of these cells perform any function physiologically. They're just there to, in the end, become these, and these do the physiological role that keeps the whole organism going. And again, you can't go backwards on this. These cells are never going to go back up over here and come down and make these cells. They're determined to go forward. So the question here is, we go back to this. What they're saying here is, at the blastula stage, if you take certain cells out, is it already? We know fate mapping where it's going to be. Right, you insert glue. You put a dye in there. You follow it through development. This is where it turns out to be. Well, at this early blastula stage where you injected the dye, was it already determined to be that, or is it going to be later signals that will tell it to be that? So that's what a lot of these explant and transplant experiments are all about. It's like, at what stage of development has that cell become induced to become what it will eventually become, and at what point is it still up in the air what it can be? You know, pluripotent versus multipotent versus it can only become certain things. So Stefan, exactly, Stefan. That's what this chapter is about. The hormones going to change that cell lineage. So what's happening at each of these steps? Hang on a sec. What's happening at each of these decision points in this flow chart is hormonal influences. One hormone will make it go this way, a different hormone will make it go that way. And so at each of these branch points, you know, and even from here to 5 different branches, it's going to be what hormones is that cell exposed to and what genes does that turn on. And that's what causes that determination. Okay. That makes sense? Questions? Okay, I was hoping that was going to take longer to talk about. Hopefully it's clear. So anyway, that's what we've been doing with these explant and transplant things. So just to define explant and transplant, explant is when you take the piece of the embryo out and you grow it in culture just by itself. That's your explant. Transplant is when you take a piece of the embryo out of one embryo and put it into a different embryo and you find out what happens. Okay, so cells differentiate along pathways to become tissues they will eventually develop into. This differentiation is irreversible. The potency, I don't know if you're supposed to use that word in that way, but I do, decreases as the cell goes down a pathway. So that 2-cell stage, both cells are totipotent, and as you split, you get less and less ability to form everything. You know, once you hit the 4-cell stage, the pluripotent, because some have got dorsal determinants and some don't. As you split into smaller things that are multipotent, okay, ectoderm's only going to be ectoderm. There's no longer any vegetal determinants after you do that equatorial split into 8 cells, right? Now it's multipotent. It's no longer pluripotent. And as you divide to smaller and smaller, each cell loses its ability to become other things, but it becomes more likely to become some kind of finished adult cell. State of determination. So that's what we're trying to figure out in these early blastula experiments. What is the state of the determination of this cell at this time? Where is it in its pathway to become what it's going to be? Okay, questions, comments, discussion? Okay, so I don't have too many slides today. I think I only cover 3 slides, but I have a lot of talking to do about them. So this is the first one. Oh, Shadarama. Do you have the tests graded? No, I do not. I gave a cell biology exam on Thursday, and then this was Friday. Almost done with cell biology, and then I'm going to start grading this one. So hopefully get started on it after this class. You guys if you guys would like buy pizza to send to my family, I would have an extra hour set because I wouldn't have to cook dinner, and I might look more kindly on your exams, if you know what I mean. No, I'm just kidding. I'm just kidding. That's bribing. That's bribery. Let me fix this. Veg tea. Okay, so what are the molecular aspects that cause the tissues that we see in the blastula? Mm-hmm. Oh, can you see this? Oh, I'm showing you I'm showing the figure, but I'm the only person who can see it, I think. So let's just, okay. So Zach Zeimer, we're looking at the Word file now, right? Give me one of these. This is yes. Are you looking at the Word file? Okay, good. Okay, just so I know where I'm at. So molecular determinants of ecto, meso, and endodermal tissue. So ectoderm is going to be determined, I think I said this before, by maternally placed messenger RNAs. So in the egg, in the brown end of the egg, there are messenger RNAs attached to the cytoskeleton that are going to make the cells that form from that tissue to be ectoderm. What are these messenger RNAs? One really important one is ectodermin. Ectodermin is a ubiquitinating enzyme. Ubiquitinating, could go either way. And I discussed this briefly last week, I think, or I forget when. I think it was a week ago Monday I talked about it. So ubiquitination is a way of degrading one specific protein. So, there's always protein turnover in the cell. Proteins become non-functional, they get broken down. But this is like specifically going out and destroying a protein for a specific reason. So we did talk about this Monday when we talked about beta-catenin. Beta-catenin, after it's phosphorylated, will get ubiquitinated, and then that will go off and be destroyed by the proteasome. Same thing here. Ectodermin is a ubiquitinating enzyme that will ubiquitinate SMAD4. So the other thing we talked about on Monday is our important signals for mesoderm induction are BMP4 and XNR, the nodals that are in Xenopus. So both BMP4 and XNR are TGF-beta-like hormones, and they both use SMAD4 as their cytoplasmic SMAD. So this ectodermin is going to destroy both pathways, basically, stop both pathways from happening. And as we'll see, these hormones are diffusing up from the endoderm, and this is going to block them, so they can only come so far. So like the whole animal cap doesn't become endoderm, it stops at a very specific point. And then another one we've got here is FOX11E. And this stops the ectodermal cells from migrating. We talked about mesenchymal cells before, which are cells that will let go and crawl and move somewhere. So in the early embryo, a lot of cells are capable of doing that. We talked about neural crest cells doing that in the last chapter. So FOX, and if these ectodermal cells crawl down towards the endoderm more, they'll get turned into mesoderm. So FOX11E is stopping that from happening. And then endoderm is also maternally determined. The bottom of the egg, the yellow part of the egg, the yolky part, is going to have messenger RNAs placed there. One is VEGT, which is a transcription factor. Again, transcription factors are going to turn on specific genes. So VEGT is going to maintain cells as endodermal by inducing other transcription factors to come on. So this is the classic transcription cascade where this protein activates these proteins and these proteins activate these proteins and like that. Happens all through development. So VEGT is going to turn on SOX17 and FOXA2, and these are endodermal determining factors. These turn cells into endoderm. So this is being maintained by that maternally placed messenger RNA. And you could, let's see, VEGT. So this VEGT is what's going to cause mesoderm to form, because VEGT will cause production of BMP4 and XNRs, the nodals, and these will turn ectoderm into mesoderm. So VEGT is the factor, the important factor that can turn mesoderm or ectoderm into mesoderm by releasing those hormones, BMP4 and XNRs. In the book, they talk about some neat experiments where if you take antisense messenger RNA to VEGT, you can get rid of all the endoderm in the embryo. So antisense messenger RNA is basically, when a gene like the gene for VEGT is transcribed, you've got your messenger RNA for VEGT. If you get a little piece of RNA that has the complementary sequence, it'll bind to it. And then there are enzymes that are going to, in the cell normally, that will destroy any double-stranded RNA. So that's an easy way to knock out some genes to find out what those genes do. What you would do is take your probably 1 or 2 cell embryo and inject messenger RNA that's the opposite of VEGT and wipe it out. And when you do that, you get no endoderm. That shows you that the VEGT is really important for endoderm. Also, you can take that VEGT, the positive strand, the actual messenger RNA that will make VEGT, inject it in the animal pole, and the whole embryo becomes endoderm. So clearly this VEGT is the factor that's causing cells to become endodermal. And again, it's doing that by upregulating SOX17 and FOXA, and those then cause the release of these hormones. Mesoderm now. What is mesoderm? I'm glad you asked. Okay, mesoderm, not maternally decided. So if you separated ectoderm and endoderm early on in the blastula, you'd end up with ectoderm and endoderm, no mesoderm. We saw those explant experiments in the last class. So it's produced from ectodermal cells. Ectodermal cells, the cells that will become mesoderm are originally ectoderm, and they are touching the endodermal cells at the margins. Okay, remember we've got the big blastocele in the middle, so ectoderm touches endoderm at the margin area there of the blastocele, but not at the top. The top of it doesn't touch at all, and they're going to respond. So these ectodermal cells then are touching endodermal cells. Endodermal cells will then release BMP4 and the XNRs, and these ectodermal cells have receptors for that. So then they'll turn on their SMADs, and they'll change, and the SMADs then will change the cell fate of the ectoderm into mesoderm. And remember, even at this stage, we've got a difference between the dorsal and what will be dorsal and what will be ventral mesoderm. So there's going to be a difference in which of these factors are produced and released where. And we'll talk about that in this class quite a bit. Okay, mesoderm induction is limited to the margin zone by the expression of ectodermin, what I talked about up there. So you've got endodermal cells releasing BMP4 and XNRs, which is diffusing up. It could theoretically activate all of that ectoderm and turn it all into mesoderm. But coming down from the top, you've got this ectodermin that's blocking the signal. So only at the margin do you get mesoderm, and you get a really sharp band of mesoderm around the middle, and no ectoderm. No ectoderm. Sharp division between mesoderm and ectoderm. Okay, so I'm going to talk about the timing in a second, but let's take a look at the figure from your book. It talks about this. So this is slide 25. Oh shit, something's wrong. Oh man, my computer's messing with me. Oh, damn it. All right, hang on a sec. I'm going to close this and reopen it and see. Your centozoals. I was singing that in cell biology yesterday. Still stuck in my head. Back to our share. Sharon is Karen, right? Karen is Sharon. Come on, baby. There we go. Okay. So what I've been saying up to now is you've got veg T down here. Veg T is making all of this into endoderm, and it's making it release nodals, XNRs, as well as BMP4. And those are diffusible hormones that are coming up this way. And when they hit the ectoderm there, they convert those ectodermal cells into mesodermal cells. And you're getting this really tight band all around the embryo because you've got ectodermin coming down from this way. And ectodermin is diffusible as well, I guess. Or, no, it's not diffusible. It's inside the cells, but it's in all of these cells. And that's going to block that movement or the induction of mesoderm. So this is cross-section through the middle of it like this. Don't forget you've got your blastocele here. So if we cut this across this way and cut all the ectoderm off, this is what it will look like. You've got endoderm that's not touching anything because it's in the blastocele, and then mesoderm all around the outside of it, okay? One end of this mesoderm will be dorsal because of that cortical rotation thing, and the other will end up ventral. So you've got dorsal mesoderm, which is notochord, middle mesoderm, which is somites, and then ventral mesoderm, which is blood, heart, I believe, and blood vessels. Okay. Let's go back to my notes. Timing. Okay. So all of this induction of mesoderm happens before gastrulation begins. Now, it doesn't mean necessarily that those are all mesodermal cells yet, but now they're fated to become mesodermal cells. They may still be showing an endodermal phenotype, but they're now fated and they will become mesoderm. So what I mean by that is like it says in 26, slide 26, we're not getting mesodermal proteins being made yet, but they will be made because it's been induced. Let's take a quick look at that. So that's kind of an interesting thing as well. So this figure, slide 26, is talking about this induction window. So these animal cap cells, these ectodermal cells, can be induced to produce mesoderm, all right? Any one of these ectodermal cells, not just the marginal ones, if you cut it out and put it in culture. So one of the things they were doing was trying to figure out the timing of this determination. So you take these animal cap cells off, they haven't been exposed to anything down here, and it turns out you can add an inducing factor, like just add BMP4 to it, or add nodals to it in solution, anywhere between 4 hours after fertilization and 11 hours after fertilization. Anywhere, well, actually 9 hours after fertilization. Any 2-hour period in this will turn that ectoderm into mesoderm. After that, if you wait till 10 hours, then you no longer have 2 hours left for that induction period, and it will stay ectoderm. So if you take these mesodermal inducing hormones, you add it to animal cap at 13 hours, it stays ectoderm. It doesn't get induced. So what these experiments show you is that the ectoderm is competent to become mesoderm between here and here. Okay, it's able to become mesoderm if it's stimulated properly. But the weird thing about that is no matter where in this period, no matter what 2-hour period you take in this, you will always get muscle genes turned on at 16, okay? 16 hours after fertilization. So while inducing these cells is turning them into a different cell type, into mesoderm cell type, it's not like the clock starts there for when they will start making muscle genes. The clock is already running for that muscle gene thing as long as you induce them at the right time. Okay? Why is that? What we'll find out later is because the induction has to do with prior exposure, which is like what's happening here, exposing the cells to these mesodermal determinants early on, turning on specific genes. Once those specific genes get turned on, the cell then goes through gastrulation, and at 16 hours after fertilization, you're at the stage of gastrulation where these mesodermal cells that were turned on back here are now facing other cells, right? They're interacting with different things because now you've got a gastrula, not a blastula. And there are other inductions going on there that are going to turn on muscle cell things. So the timing of it then is all based in the timing of the gastrulation and neurulation and everything. I don't know if that made any sense. I hope it did. It was just a paragraph in the book that I thought was really important. So there it is. Any questions about that? Is that confusing? Sometimes when I think I'm confusing, I keep talking and repeating myself, and because like if I was in front of you, I'd be looking for feedback. I'd be looking for that face going, really, Dr. Slish? Or what? And I could figure out what you guys are thinking and report, change what I'm saying. Are there any questions about that before I go on? Nobody? Okay. Well, let's continue here. So kind of where I was getting into with this was position. You induce, but then its final disposition based on its position in the embryo, because it's going to come up against different tissues and become something else when it does. So this is what I just said. There's a window of time when the ectoderm is competent to become mesoderm, between 4 and 11 hours. So you need 2 hours somewhere in that period. And the induction has to occur in that window or else it's not going to happen. Mesodermal proteins are made during gastrulation, which is 16 hours later. And the timing of this is not dependent on when the induction happened. That induction can move. So there's a separate clock for the two. Okay. Bum, 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 bum. Zygotic transcription. Zygotic transmission. God damn it. Zygotic transmission. So what this is talking about now is when do the nuclei in the embryo actually start transcribing new genes. Like I said, almost most of your proteins, almost all of your proteins made up through the mid-blastula stage are all made from messenger RNAs laid down in the egg, maternal RNAs. And when you get to this mid-blastula transition, suddenly transcription starts happening from the new nuclei that have been made. And it always happens, this mid-blastula transition is after the 12th cleavage, when you've got a little over 4,000 cells there. So at this stage then, transcription starts from the zygote nuclei, zygotic nuclei. And at this point then, all the cell divisions start to become asynchronous. Talked about this in chapter 3, how, at first all the cell divisions happen at the same time. Then at one point, the ectodermal cells start dividing faster than the endodermal cells, and then epibole happens and it covers the whole embryo with skin. So this is when that starts to happen. Once the individual nuclei are activated, then they don't necessarily divide with the whole embryo anymore. And inductions at this point start to change cell types, right? You can't have an induction unless you can turn on new genes. So that's what's going on there. Transcription is the reason why. The reason why it takes until the mid-blastular transition, they think, is that maternally there's laid down a transcriptional repressor. So there's a protein there that's like a general repressor that's going to turn off all transcription in the egg until the egg is ready to start transcribing, that mid-blastula stage. And so the idea is here at this early stage, you've got a certain concentration of transcriptional repressor here. And you've got one tiny little nucleus, so you've got a lot of repressor protein around. Now, through these early divisions, you're getting mitosis, which is division of the nucleus without any cell growth, right? So the individual cells are getting smaller and smaller and smaller and smaller. So the ratio now between the size of the nucleus and the size of the cell changes dramatically, right? Here you've got a tiny nucleus and a huge cell. Here you've got the nucleus filling up most of the cell. And so the amount of DNA per cytoplasm is increasing as well. So if you've got a constant concentration of repressor protein in the cytoplasm, and then at the beginning here, that repressor protein can bind to all the DNA, stop all the DNA from transcribing. As you go on, you start to dilute out that repressor protein because you've got more DNA that it can bind to. And at this point, you've got more DNA than there is repressor protein. And so now the new genome can be turned on, okay? So there isn't enough repressor protein at this point to shut all of the genes off, so now genes start to get turned on. And this is what they're trying to stay over here with this egg timer thing. You can say this is like fertilization, and as the cells divide and divide and divide, the concentration, the ratio between DNA and repressor protein decreases till you pass this threshold. And this event is that mid-blastular transition. And then from then on, you're getting transcription from that, zygotic nucleus. Okay, makes sense? Mid-blastular transition, that's when you start getting transcription from that embryo's actual nuclei. Bum, 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 bum. Okay, one more thing I wanted to talk about today, and that is positional information. So, do I have a slide on this? Does not exist. So, if we go back to slides 19 and 22, something we talked about before, what will we see? I'm curious to find out myself. I wrote this earlier today. What the hell are you talking about, Dr. Slush? So we know at this stage, at this point, there's really no mesodermal tissue because we've induced it, but it hasn't produced any mesodermal proteins. But we know this is where the notochord is going to end up, right? So its position is really important. The position it is in the embryo is really important. And we showed that again here in slide 22, where if you take this chunk of marginal zone, it will become dorsal mesoderm, and this chunk will become ventral mesoderm. So the position it is in the embryo is really important to what it's eventually going to become. I think that's all we're talking about here with positional information. Let me read the notes again. Okay, so the dorsal ventral patterning is already determined in that blastula, even though you don't have a dorsal ventral axis in the blastula. So that'll be the DV axis will happen during gastrulation, but the cells that are going to be dorsal and ventral are already determined. Different parts of the endoderm have different mesoderm inducing abilities. So we're going to talk about this and why that is. It has to do with your BMPs versus XNRs that are all being produced by the endoderm. And an endodermal cell from a putative dorsal side will induce ectoderm to be-form dorsal cells. And endodermal cell from the other side will make ectoderm into ventral mesoderm. Let's take a quick look at that one. Oops. Okay, so if we take endodermal cells from here at this blastula stage, and we take the animal cap and we put them together, we're going to get dorsal mesoderm. If we take vegetal cells from here and the animal cap, we're going to get ventral mesoderm. So it's not the ectoderm that's determined, the position of the cells in the ectoderm that decides it. It's the position of the ectodermal cells with respect to specific determinants within the endoderm. And, you know, I keep saying this is kind of this is kind of a historical way they figured out all this stuff. I've already told you the answer, right? We know why that is. It's because that cortical rotation forced the dorsal determinants over here. So in these cells, you already have expressed a lot of beta-catenin. You probably also have siamois upregulated in these cells. So when you pull these out, these cells are going to make dorsal. These cells are going to make ventral because they don't have those dorsal determinants. Bum, 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 bum. An endodermal cell from the other side produces mesodermal cell. Take home message. The position of the marginal cells is important in determining the fate of the cells. Okay. So that, my friends, is what I have for you today. We're about 15 minutes short. Is there any questions? Maybe we'll have a quiz on Friday to use up some more of the space. Michelle, were you going to say something? Michelle Simmons?
Oh, no, sorry.
Oh, you lit up in yellow there for a second. And I'm anxious for at least someone to speak. Someone, talk to me, talk to me. How about Audra Santonge? How's the class going for you, Audra? See, she's not there. Just as I feared.
It's horrible.
It's horrible. Vanessa, come on, man. I know it's hard. It's conceptually hard because you have to imagine 3 dimensions from 2-dimensional pictures, and it's hard, but stick with it. Keep reading it. Keep looking at it. Make sure you're reading the book.
No, it's not that bad. I was kidding.
Oh, don't lie to me, girl. Okay. Any other questions or comments? Discussion points here? Nothing? All right, well, why don't we call it a day? And I will hope to see you all back here again on Friday. Later, dudes.